Dr. Father Nick Estriaco.
That's correct.
Okay. He is an associate professor of biology and an instructor of theology at Providence College. And then once again, his title is up here. And after all these problems, I hope it all
Thank you
works.
so much.
You're welcome.
So I'd like to begin by thanking Phil and Carter for the kind invitation to come and speak here. I was telling a couple of people at lunch that I've spent the last 5 years basically getting tenure as a biologist at Providence College, running a laboratory that looks at programmed cell death in yeast. And so this is in a sense, the first opportunity in a long time to return to these issues and to talk and to think about the relationships between the biology that I love, as well as the philosophy and the theology that I was introduced to later in life when I discerned my vocation to become a priest, a Dominican priest. So today, what I'd like to do is I wasn't quite sure who was going to be in the audience. I had hoped to be here this morning, and usually as a biologist, you go to the meeting, you listen to what people are talking about, then you adjust the PowerPoint appropriately to what's going on. And I'm sure the biologists here know what I'm talking about, but I was stuck with Christopher Tollefson in Detroit's wonderful international airport. So, we've discovered, it's a huge airport, and it's just a wonderful place to just walk around. So, unfortunately, I wasn't here to be privy to your discussions, and I hope that I won't be repeating some of the things that you've talked about. My goal this afternoon is to try to bring together some of the biology, especially the biology from a systems perspective, from systems biology, with some of the classical notions of matter and form. And my goal is to try to, in a sense, illuminate what goes on in life, in the living organism, because before we do bioethics, and that's what I do with the other half of my brain, before you do bioethics, you need to talk about what you're looking at. And especially in the past few years, much of the debate has been, is this in fact, and this you name it, it could be an altered nuclear transfer from hurl, but what is it exactly? And before you can talk about whether or not you can do something with it, you need to talk about what it is. And so this project, in a sense, this research project was started while I was still in the seminary preparing for the priesthood during my philosophical and theological studies. And so now it's a wonderful opportunity to return to that. And so the pressing question here, of course, is what is an organism? And from what I understand we had a brief conversation with, is it John? James. And he was very good. He was like a reporter. He gave us a sense of the flavor of the conversation at the workshop this morning when he picked us up at the airport to drive us here. And I know that for biologists, we usually don't think about this. I mean, we spend so much time learning the techniques, running a PCR reaction without getting any contamination in there, that we don't really think about the philosophy involved. And so what I would like to do is try to engage those questions using biology to answer that. And so we're going to talk about what is an organism. And to highlight the import of this question, I would like to go to a question. I don't know, all of a sudden we've got these shadows, but I apologize for that. I guess in the translation between Macs, we've got shadows that show up. But this is the infamous, famous ship of Theseus problem. And it goes all the way back to pretty much 2,000 years ago. And the question is the following. You have a ship. It leaves Athens. And over the course of its journey on the ocean, they replace every single part of the ship with a new plank or a new bolt or a new rope. And it comes back. Is it the same ship? And it's a wonderful question to throw out to freshman students. And it deals with questions of identity, it deals with questions of change. And the question is, did the Athenians still have the one and the same ship? And you can take this problem and make it more complicated. You can have a situation where you take the planks as you replace your ship, put them aside, and when you get back to Athens, you build a new ship. So now you've got two ships. The ship with the original parts from the ship of Theseus, and then you have the ship that has been gradually replaced. Which is the original ship? Are there now two ships, one ship? How do you deal with that type of question? Well, I'm going to leave that to freshman philosophy. But you see, it's an important question because, in a sense, something is similar happens with us. And so And this is decades of kinetic and metabolic studies. And I've summarized some of that data on this slide, but I'm actually not going to go over the data. I'm just going to highlight the main conclusion is that 98% of the atoms in the adult human body, including those found in the brain and the nervous system, so the nerves actually remain, but the atoms that make up these cells are in dynamic motion. They're always changing. So basically after 650 days, plus or minus a month or so, every 2 years, what happens is you've got new atoms, except for the ones in your skeleton. In your skeleton and the heavy metals in your body, those are pretty stable. The question now is, are you the same human organism you were 3 years ago? Or have you become someone different? Are you someone new? Now, obviously, common sense says that, yes, this person was, in fact, this person 70 years ago. And yet, we need to talk about what that means, given this biological observation. Is there an ontological difference between ships and human beings? A very simplified philosophical question. Or, to put it another way, how do we explain the identity and the continuity of you and me? That's really the question at the heart of the project that I'm attempting to think through. And I'm appealing back to hylomorphic theory. And hylomorphic theory, I'm assuming that you've talked about it, you've argued about it, you've gotten confused about it for some of you this morning. Hylomorphic theory, basically from Aristotle, posits that to explain this continuity and change. And you see, it's a theory, it's an explanation, it's an attempt to explain how you can be who you are despite all the changes that you go through. This is a theory. It says that all living things are substances, and they're composed of a formal and material principle. And I'm going to, picked this word as a technical word in philosophy, but I'm going to hold on to that for those of you who are not philosophers. And both form and matter are inseparable in material things. And I'm now bracketing away the unique case of the separated soul. I'm not going to go into that question. We now then talk about what a form is. So by definition, a form is what makes you what you are, as in a human being versus a dog. And it gives you specific causal powers. So dogs bark, we don't, unless we try to be dogs. And the ability of trying to be a dog is something that we have. And so those particular causal powers for the classical tradition can be attributed to form. And then you have matter, and matter is stuff. And one of the difficulties when I teach this to my undergraduate students is they have a post-Cartesian understanding of matter. This is matter, and then you've got energy. And we have to refrain from that. So if that's what you think about when you think about matter, stop thinking about it. Because matter, understood classically, is not exactly that. It is the underlying stuff, and I'm going to try to talk about that a little bit more as we move along. Now, when a lot of people, this is supposed to be a triangle. So a lot of people, when they think body and soul, they think Descartes. You've got a body, and you've got a soul, and somewhere along the way they touch. Descartes taught it was the pineal gland. And so a lot of people working on the philosophy of mind today are trying to work out, if they are dualists, how you have this conversation between something that is spiritual, the soul, and something that is material, the body. And hylomorphic dualism is much more profound than that. Because you see, hylomorphic dualism says that it is so intimately bound together. That the material principle and the formal principle are so tightly bound that rather than talking about a soul and a body, you have to talk about a body-soul composite, where the removal of the soul does not leave a body. Now, this is, and if I had a class here, if this was Intro to Philosophy, we'd go into the implications of that. The idea is that the human corpse after death is not a body properly so called. It's basically the atoms that are left behind once someone is dead. Because the soul is the principle that organizes the body. It makes the body what it is. So in the absence of a soul, there is no body. You just simply have stuff that needs to be organized per se. Now, I'd like to highlight 3 things about classical hylomorphic theory and then hope to try to show you how we can use systems biology to recapitulate these principles. So first of all, it's substantial. So if you talk to Aristotle, if you talk to Aquinas, they talk about substances. And I know there is an entire debate amongst the analytical philosophers about the very nature and existence of substances. And I'm going to, again, just not deal with that at the moment. They talk about substances. Substances are real, and they can be distinguished from artifacts. So we are substances, boats, wooden boats, ships are artifacts. It's also holistic, so the whole is real. So you and there's something real about you and me. There's something different from you and me and a 70-kilogram collection of atoms. And this whole has properties, it has dimensions of it that make it different from simply the combination of all the parts. And finally, it's dynamic. Change is real, but persistence is real too. You change constantly. We just had lunch. We're changing. And yet, we're still the same people who went into the Morrison to have lunch. And so this dynamic relationship between the persistence of the person through continuous change is part of the hylomorphic theory and can be attributed to its notion of change. So what you have is that you have form and matter, and that allows you to say that change involves replacement of form, but the endurance of matter guarantees continuity through that change. And that's why actually if you look at Aristotle and Thomas, you have these 2 correlative principles precisely to explain change. Now then they talk about 2 kinds of change. There's substantial change, where you involve the replacement of a substantial form. And what's key about this philosophically is it radically alters the identity of the thing. You do this once when you die. That's when you radically change. Now, we do this all the time, accidental change. We call this gaining weight and dieting, amongst other changes. What happens here is you have replacement of accidental forms where the kind of change alters aspects of the thing, and the thing is able to perdure through that change. And so we're talking about that. Now the key is, how do we talk about form and matter in the 21st century, especially to my colleagues who are biologists? I mean, that's really the challenge. I have friends back at MIT where I did my PhD, and we had wonderful conversations about form and matter, about God, about life, about everything. And part of this is an effort to talk to them. How can I talk to them about these fundamental principles from the philosophical tradition that are so important to understanding, especially if you are a Christian, especially if you're a Catholic Christian, the whole worldview that is presupposed by that tradition. So we'll talk about systems hylomorphism, and it has to deal with systems biology. And in order to do that, I need to talk about a cell and the fact that cells are made up of molecules. Now, for those of us who are biologists here, that's intuitive. But for an entering freshman biology major, that is not. That, in fact, you and I and all living things are a dynamic dance of molecules. And so the best way to do that is to show you what I show my freshman students. And this is a really cool interior view of a living cell. And what you're going to see captures the molecular changes that happen in a white blood cell as it stops circulating in the system and begins to change its biology so it can basically enter the tissues and run after some bug that it's about to encounter. And so this is a BioVisions from Harvard, and I'm hoping that we'll be able to see it. So let's see. I don't know if there's volume. My students love the music. I'm going to stop there. Okay. What I wanted just to illustrate there was the fact that the living system is made up of incredibly dynamic interactions amongst different molecules. And here is my favorite. This is kinesin, and this is a motor protein that's basically running up and down. Well, actually, it's running up. It's running up one of these fibers, and it's basically carrying a vesicle. But what I wanted to highlight is that the cell is made up of molecules, and they're interacting with each other. And they're interacting with each other in very specific ways. And so they're systems of molecules, and they're organized in a particular way. And different cell types have different subsets of expressed genes. And what I mean by that is that you can tell one cell type from another precisely because there are different molecules that make up cell type A from cell type B. And I took these figures from a paper that was published in the Journal of Immunology just last month. Here you've got a diagram, it's called a heat map, of the molecules that are present in the T cell versus a B cell. And the idea is that you have a list. You can't see them, you can't read the list, but that's not important. The idea is that each one of these names indicates a particular gene. And the color, red or blue, tells you whether or not that gene is on or off at a particular given point in time. And what these two figures want to show you is that T cells, at this point in time, and I'm not going to go into the particular experimental details, they are red here versus B cells, which are blue. B cells are red on the left. They are blue on the left-hand side for T cells. And so T cells and B cells can be defined by the subset of genes that are turned on in those cells. And in fact, you can identify a particular cell type simply by reading a list of all the genes that are turned on in that cell. And so, and I think you had an opportunity to look at the paper where I first described this. If you have a system of a particular constellation of molecules at time equals 0, what ends up happening is that these molecules interact and they move along in a particular defined trajectory, such that what happens is the inherent physical and chemical properties of the molecules drives the living network. And so the idea here is that if I want to know what something is going to be at time equals t equals 10 minutes, I should be able, by looking at the interactions of the molecules that are present at time equals t equals 0, in a sense predict what's going to happen. Now, I'm again, clearly we have to drink, clearly we have to eat, and so there are molecules that are entering the organism, and the interaction with the organism is very dynamic. But the idea here is that a particular cell or a particular organism, I'd like to point out, from the molecular composition of that cell or organism, you'll be able to determine its cellular behavior. And if you want to change, if you change the molecular composition, you actually change the cellular behavior, and vice versa. If you have changes in the cellular behavior, they come about because of changes in molecular composition. Now, this is a paper published in PNAS in 2010. So here you have stem cells. And what you do is you take the exact same stem cells and you put them in a container that is shaped like a flower or is shaped like a star. And simply the organization of the stem cells within that compartment determines their eventual differentiated state. So flower shapes make fat cells, and star shapes make bone cells. So it's a striking illustration of how the architecture of the molecules determines the particular outcome that's going to happen. Now, different organisms, too, have unique signatures. Here's my favorite organism. This is Saccharomyces cerevisiae. And here you have a map. It's an interactome. It's actually called a hairball. And a lot of people have pointed out that where the double helix has become the symbol for molecular biology before the genome, after the genome, it's going to be these hairballs. Because these hairballs really emphasize the holistic nature of organisms. So here you've got all of the interactions that are present in budding yeast. This is the human being. This is St. Thomas Aquinas, my very famous Dominican brother and theologian. And here you have the network that defines the human species. And so what you have here is, in a sense, a species-specific organization of molecules that defines what a yeast cell is. And this defines what a human being is. And my suggestion is that if you want to talk about form and matter in the 21st century, you have to have that background in mind. You have to point out that here, what an organism is that it's a molecular matter organized in a species-specific network with its particular telos. And telos basically means an end. And so you have these molecules organized in this way, and this defines human being. And these molecules organized in this way, that defines yeast. A form, it basically, if you ask what is the form, well, the form is the underlying reason for that organization. So if I ask the question, why are yeast cells like this and human cells like this, the philosopher will say, well, the yeast form is the explanation for this, and the human form, the soul, is the explanation for that. And so you have a way of talking about form and matter using the insights given by biology, by systems biology. Now, when we talk about stuff, again, it's not the molecules themselves. It's the most underlying constituent of stuff. And people have equated matter with quarks, with strings. I mean, the most fundamental elements of nature organized in a particular way will give rise to an organism. So let's talk about how we can talk about the substantial perspective. So here, molecular networks are real as well. You can actually see the whole network. The holistic perspective, now we're not simply talking about this particular protein. We're looking at this particular protein within the entire context. And we have proteomes and transcriptomes and metabolomes. And now in systems biology, there's this emphasis on how the individual molecule has to be studied within the context of the network in order for it for you to really understand what it's doing. And I believe it was actually here at Notre Dame, the whole systems perspective with Barabasi in physics. Is that how you pronounce his name? I guess he's now moved to Barabasi. He's now moved on to Northeastern. But he was the pioneer at really defining the contours of living networks. He's actually looked at Facebook. And what's striking is that Facebook, the structure of Facebook, very much resembles the structure of a living organism. Now, and we talk about the dynamic perspective too. We can now try to understand change. Now, what is development? So the readings this morning focused on development. We're trying to understand what development is. Well, from the systems perspective, it's an unfolding of a process that's defined by the intramolecular reactions of the network established at fertilization. So once you fertilize the egg, and this is what's unique about fertilization, right? So the egg basically has a lifespan of only about 24 hours. After that, it depletes its resources and it dies. But once you have fertilization, once the calcium wave is initiated, what ends up happening is that system changes. And that system changes, it begins to develop. The molecular mechanisms that come into place at fertilization develop, and you eventually have a continuous motion until you die. Now, what's interesting here is that chicken embryos do not become dogs. And the reason is because the intramolecular network established in a chicken embryo is not ordered towards dogs. It's ordered towards adult chickens. And so here, philosophically, we could understand why the final end is established by the formal cause. And this one is a philosophical claim. And what I'm trying to propose here is that there is a biological basis for the philosophical claim that was made by Aristotle some time ago. Now, what about change? We talked about there's substantial change and accidental change. Now, what's important to note is that living systems, just like Facebook, is organized in a hub-spoke system. This is Atlanta, and that one's Detroit. And if there's a lot of storms over Atlanta, the whole thing dies. And Dr. Tollefson and I get stuck because basically Atlanta shuts down, and the planes can't travel through that hub. And so airplane networks are hub-spoke systems, and Barabasi showed that living networks, like the network I showed you for yeast and the network I showed you for humans, are also organized in this way. Now, what's striking is, according to a paper by Zhang et al. From the Vidal lab at Harvard, what ends up happening is that if you have essential genes, more often than not, they code for hub molecules. But if you have non-essential disease genes, they often code for spoke molecules. And so the proposal is that, can we talk about substantial change as changes that involve molecular changes in the hubs, while accidental change is actually molecular changes involving the spokes. And so here you have a system where, again, it's an attempt to translate a philosophical framework into contemporary biology. Now, what sort of, talking about stem cells, this is a stem cell workshop. When you talk about a pluripotent stem cell, this is the standard definition that's usually given. It's a stem cell that's able to differentiate into all the somatic cell types of the adult organism. This is a functional definition. I'd like to suggest, in light of what I've talked about, it's a living network of molecules driven by OCT4, SOX2, and NANOG. And I'm referring here to a paper from the Yanish lab as a review in Cell, where they point out that OCT4, SOX2, and NANOG, in a sense, are the core regulatory proteins for the entire thing. So once, and this is where reprogramming comes in. I mean, reprogramming is basically an attempt to jumpstart the three endogenous molecules. So when they become present, they take over the entire cell and reprogram the entire cell. And so could we not define a pluripotent stem cell, again, based on this network idea? And here, of course, for those of you who are not familiar, and from this morning's talks, and I'm sorry I wasn't here, but I think Rebecca brought this up, where you're able to simply reprogram them by initiating, by stimulating the expression of these 4 genes. Now, these are the questions that I'm just going to raise, the questions that I'm thinking about. First of all, how similar or different are the species-specific networks found in individual members of that species? And this is the question of developmental noise. Part of what I pointed out here is that we've talked about, say, the network for yeast. But there are different yeast cells, some in my lab, some over here at Notre Dame. What happens when we start looking at individuals rather than simply the universal, philosophically? And then how are we to understand the development of chimeric organisms? And this is the very famous GEEP. So a GEEP is a goat-sheep mixture. And you basically have an organism that's part goat and part sheep. Now, how does that work? I don't think, I mean, especially from a network system or system, how does that actually work? That's sort of stuff I want to talk about. Because now, if you have 2% sheep and 98% goat, what does that mean? Is that all goat? Is that just a goat that happens to have sheep cells? When do you actually have a GEEP? Is it 50-50? These are the kind of questions that I think are important in light of the type of technology that we can now do with embryos, that we can do with organisms when we start mixing and matching. And if we're going to be talking about whether or not it is moral to do X or Y, we need to be able to understand what exactly is being done with what and what the final product is final product is made. So I'd like to talk, this is my lab, and I told them that I'd end my talk, as I end all of my talks for the past 5 years, thanking them. Because one of the great joys of teaching at Providence College is my undergraduate students are also philosophy. They have also taken philosophy. So the conversations that we have deal with knocking out Bax inhibitor in yeast and looking at the phenotype of that mutant. At the same time, we talk about the brothers Karamazov, as well as talking about the nature of human organisms. So these are my students who've worked with me, and they have been the ones who've asked the questions that stimulated the talk that I gave to you today. And this is the funding that has kept me busy and my lab busy for the past 5 years. Thank you very much.